Good afternoon. I know you all are here after lunch. I'm sure some of you may snooze, and we'll try our best not to give this kind of an opportunity. But to start with, you all know what retina is? You all know what retina is? Yeah? So it's like a screen on a movie theater. Yeah? The optics of the eye makes the vision focus on the retina. So the brain processes and gives us the real world picture. So there are common eye diseases like age-related macular degeneration or retinitis pigmentosa, where majority of these patients cannot see because the cells will not be able to regenerate in these diseases. So here we are with Professor Masayo Takahashi from the RIKEN to give us the recent updates on different treatment modalities in case of retinal cell therapy using Ips cells. Ladies and gentlemen, it's my great pleasure and privilege to welcome you all to this distinguished lecture at the Auditorium of School of Life Sciences, University of Hyderabad. And I take this opportunity to extend a warm welcome on behalf of School of Medical Sciences, University of Hyderabad, and welcome all special guests, friends of the university, and also the students. And we honor this afternoon in the presence of our Dean, Professor Prakash Babu, and Dr. Geeta Vemuganti, and the VC, Honorable VC will join us soon to the dais. And may I take this opportunity to invite the distinguished speaker, Professor Masayo Takahashi, and Professor Prakash Babu, and Dr. Geeta Vemuganti. So before starting the session, would like to request the Dean, School of Medical Sciences, Professor Prakash Babu, to give us a welcome address, please.
Very good afternoon, all of you. Today's speaker, Professor Masayo Takahashi, and my colleague, Professor Geeta Vemuganti, and my colleagues in the audience, participants, and invitees. It's a great pleasure to have such a kind of wonderful talk by Professor Masayo. So today morning we had a one talk by the Nobel laureate in the morning at 11 o'clock. And this is the second talk today. And this has some uniqueness in the talk, as you have been listening about the stem cells and their transdifferentiation and their possible repair. However, so now the recent trend is the IPS, induced pluripotent stem cells. How you can modulate them towards a particular lineage and how they can be repaired and all. And what I understand from Professor Masayo, they have been associated with the RIKEN and this research. RIKEN Institute is one of the renowned institutes, research laboratories in Japan, and they have been doing very wonderful research, especially in the neuroscience. And so this topic is a really interesting one, even to me, when she said that the retina is part of the central nervous system, extension of the central nervous system. And though we teach up to optic nerve or up to cribriform plate, where you will have the sensory nerves up to the nasal, but we did not touch the retina, we always feel that that's not our area. But so you enlightened us. And also, as my colleague Dr. Nagaraj explained, there are several things associated with the eye. And without that, I don't think anybody can do anything. So this is an important sensory organs. So this is the very good research work, so that the regeneration of that level is an important aspect for especially the developing countries, where children are born with inborn errors as well as with accidents. So this research will have the real application and bench to the bedside and clinical trials. So with these few words, I welcome all the guests and participants for this. Especially I extend my warm welcome to the today's speaker, Professor Masayo. So thank you very much.
Thanks, Professor Prakash Babu. Now I would like to invite Dr. Geeta Vemuganti to introduce the speaker. Thank you.
A very good afternoon to all of you. And it's my privilege and honor to invite Masayo Takahashi to University of Hyderabad. Actually, it all started, I had extended an invitation to her 3 years ago, and after I moved here, I said, you should visit University of Hyderabad. She said, I will do that, and anytime I come there, I'm definitely going to touch upon you. And when she was invited to the Shampili Mode Foundation at L.V. Prasada Institute, and she sent a mail that she would come and visit me, I said, that's not enough. And it would be an honor to have her distinguished lecture here. And it's, again, as I said, it's a privilege to me to introduce her because I know her professionally as well as personally, and it's really an honor to be associated with her. Masayo, as we call her, she's now the project leader at the Laboratory for Retinal Regeneration, RIKEN. You must have most of the activities on these stem cells, IPS, the clinical trials come from that area. RIKEN Centre for Developmental Biology, Kobe. And about 8-9 years ago, I visited her lab. And when you go to each lab, you find the science papers, nature papers on their boards. And that was one of the most impressive thing when I visited their lab. And we had a lot of interaction with all their colleagues. And Masayo first did her, though she's now in the project, she's both from the science background as well as from the medical background. She has done her MD from Kyoto University, Japan in 1986. And then she pursued her PhD in medicine at the same institute, which she completed in 1992. So another rare breed of clinician scientists. And she served as an assistant professor in the Department of Ophthalmology, Kyoto. And then she went to California, and that's where I think that was the first time you got into the stem cell research. And at the Salk Institute, she got interested and she got trained in the stem cells. And subsequently, she came back to Japan and started as an associate professor at RIKEN, and she developed the team to start with retinal regeneration. And she initially started with the pilot. How many of you are familiar with IPS? Okay. So I would just make it little interesting, those of you who are in the stem cell biology. 20 years ago, we thought that embryonic stem cells are going to do everything for us. They can regenerate every organ. Then came the ethical issues with the Bush government putting a ban on stem cell research. We all focused our attention to mesenchymal adult stem cells. Then we realized that the limited potential of the mesenchymal stem cells for plasticity. Then why not look at embryonic stem cell again? And that's when the combination of adult stem cells with 4 genes of embryonic stem cells, and that is thanks to the two Nobel Prize winners, Yamanaka and Gordon. They came up with 4 genes which can change an adult cell into embryonic stem cells. So we couldn't use embryonic stem cells, but we have embryonic-like stem cells derived from adult cells. So those who have been following the stem cell biology last 15 to 20 years has been very exciting. In a conference called Association of Research in Vision and Ophthalmology, ARVO in short, the latest hot topics in research are discussed there. And that particular hall is flooded with people, and they're all overflowing into the corridors. And we remember the first time that the IPS was discussed in ARVO. It was so exciting, we started talking about it in ICSCR, the Society for Clinical Therapy, Society for Stem Cell Biology. Every stem cell meeting had this topic of induced pluripotent stem cells. And now we'll hear that it has come to a stage where you can derive some of the cells which have been proven to be functioning as adult or like the adult cells. So from the IPS, so we took 3 steps backwards to make adult cells into embryonic-like cells, and now we are pushing them into the differentiation that we would be interested in. So, and Masayo has mastered this and her lab, and she would talk about her work. And not only she's a fantastic retinal surgeon, and this is a rare opportunity for her, that she grows the cells in the lab, and she has an opportunity to go and transplant it into the patient along with her clinical team. And that's an honor and privilege that she has. She worked for it. And so she would be talking about the first transplantation that she did, which is NEGM, what we call it as Makkah Madina of the journals for medical people, the New England Journal of Medicine. She published her work there, and she would be now talking about the other work or some of the patients that she has transplanted. She has more than 200 publications, and her H index is 64. So with this brief introduction, I once again welcome Professor Masayo Takahashi, and thanks to the Dean School of Medical Sciences who has taken out his time. And on behalf of the Vice Chancellor, I convey that he had another meeting, and he said he would be joining because of his earlier commitments. He's not able to make it here. So with that, I would now invite Professor Masayo Takahashi to deliver her lecture. Thank you very much.
Okay. Okay, thank you. Thank you for the wonderful introduction. And it's a really honor for me to talk about my project in here, the same famous university. And it's a great pleasure to meet Geeta again, the cheerful and always kind person. So I really enjoy visiting, the first visit in India. And I will talk about the retinal cell therapy using iPS cells. This is a translational research, so not much science. And you can see that for the translational research, you don't need much brain, but the toughness you need. So please be relaxed and listen to my talk. And I'm relieved that many of you know about the iPS cells. And Geeta introduced very well, but this cell sheet, retinal pigment epithelial cell, that is a part of the retina, that is derived from the patient's skin. So skin turn into the retinal cells. It's amazing technology. So my first research began at the 1995 in the Salk Institute, where I learned about the concept of neural stem cells. And Dr. Gage's lab, he succeeded to culture the adult brain-derived neural stem cells. And at that time, the concept was not accomplished, and not many people believe in the neural stem cells. But I was really lucky. I'm an ophthalmologist and went to another field, basic brain research laboratory, and I encountered the new concept of neural stem cells. So I thought, gee, I'm the only one in the world who knows about the neural stem cells as an ophthalmologist. And quickly I applied it to the retina. So until then, I was 35. Until then, I didn't think anything. But suddenly I encountered my whole life theme. So don't worry if you don't have anything so interesting now, but in the future, if you are doing something vigorously in one field, and then if you look another field, you will find a very good, interesting thing, and the new thing will happen. And in Gage's lab, actually, I just followed my husband, and he proved that the neural stem cell is really stem cells, because people doubt if this is neural stem cells. And the definition of the stem cell is the multipotentiality and the self-renewability. And he proved, first, he cloned from the cluster of the hippocampus culture cells, he cloned one cells and expand them and make them differentiate into various cells. And he found one cell line can differentiate neurons and astrocytes and oligodendrocytes. So this means the first cells have the multipotentiality. But to prove the self-renewability, you should subclone again from the proliferated cells and prove the multipotentiality again. Then you can tell the first cells have the self-renewability and the proliferated cells have the same character. So the neural stem cells was proved at that time. And I applied this concept to the retinal transplantation because at that time, Dr. Das in India did the fetal retinal transplantation. So I knew that retina needs some neurons. So I applied the neural stem cells. Even though that was a brain-derived neural stem cells, I applied that to the retina. And at that time, I say to the boss, Dr. Gage, I will transplant the neural stem cells to the retina. He laughed a lot because from the aspect of the brain, he didn't think retina is a central nervous system. But I believe that this will make a new treatment, completely new treatment. And I found the neural stem cells integrate very well, and the morphology was like retinal cells like this. But after 6 months, I found these are not the authentic retinal cells because it's from the brain, hippocampus. But at that time, we thought the neural stem cells from various region is the same because the morphology is same. But nowadays, it's common sense that the neural stem cells from various places has different characters. So, but at that time, I thought, okay, I can make a treatment from this, and I thought easily, but the things going not so easy. And from that, my journey began, and I went through all these steps: basic research, application research, translational research, and now I'm in the stage of clinical research. And soon we will go into the clinical trial. And please note that the basic research and the application research, the using tool is same: molecular biology, animal experiment, and the cell biology, but the aim, direction of the aim is completely different. The basic research, you will seek for the truth of the science, but the application research, we will seek for the method. So we always think how. So completely different. And so after this talk, don't ask me why. Okay, you can ask any serious or severe question, but don't ask me why it is so. Okay, and each step is so important. Usually, it is thought basic research is more important. Of course, basic research is important because otherwise, nothing new will come out without any basic research. But also, application research and the translational research is very important, and it will take 5 years or so, each step. And during the development of this treatment, I was told many things. Retinal regeneration, it's central nervous system, so it's impossible. Or the ES cells can be used for treatment. Or even now, I'm told iPS cells are too dangerous. Don't think about the clinical use. But I was not persuaded because I knew from the neural stem cells or genetics to the disease and the clinical treatment, surgery. So I believed that we can make a new treatment with the stem cells. And when I came back to Japan, I soon realized the somatic stem cells, neural stem cells, cannot be expanded for enough amount to treat the many patients. And if we expand them, they will change the character. So to make the treatment, I moved to the ES cells because they proliferate and they can make any type of cells in the body. And Dr. Sasai showed me this plate one day, and he told me that I'd like to make the dopaminergic cells from ES cells, but I found these brown colored cells, and please check if this is a retinal cells. And immediately I saw these dishes. I think we can use this for the age-related macular degeneration that caused by the senescence of the retinal pigment epithelial cells. At that time, this disease is not so famous. So even though Dr. Sasai is a top-level developmental biology researcher, but he couldn't think of the treatment of the age-related macular degeneration. But combination of the basic researcher and the clinician immediately find the way of developing treatment. And I proved that primate ES cells can treat some animal model of the retinal degeneration. And that was the first report using primate ES cells to treat something. But I was hesitated to go into the clinical scene. That was a almost preclinical stage, but I hesitated to go into clinical application because for the age-related macular degeneration, it's a small lesion in the retina in elder people. So if we use the ES cells, we should let them take the immune suppressive drugs. So that will harm the elder people. So I was hesitated. And then the IPS cells came. So using the IPS cells, we can make the retinal cells from patients' own cells. So that will clear the hurdle, the last hurdle. So I quickly moved to the IPS cell research. So I was told many things. It won't happen, but I was doing the research so that I can receive the progress of other field. It's very important to do something, believe in something and doing actually something. Okay. And ES cell and IPS cells is the character is the same if we culture them properly. If you culture them roughly, it tends to change the character and went into the very bad IPS cells. But if you maintain the IPS cells very gently, they show the same character as ES cells. And as Gita introduced, the IPS cells is made from the somatic cells of adult, originally from fibroblast, but nowadays we can make it from the blood cells. And in 2006, he made the mouse IPS cells, but 2007, human IPS cells came out. So from that point, we were at the preclinical stage with ES cells. So I thought I can do the clinical treatment within 5 years because we just clear the preclinical stage. And I said so. Nobody believed that, but I said so. And at that time, the regulation is the limit of the clinical application. But IPS cell has much power in Japan, especially in Japan. Amazingly, 86% of the whole population in Japan knows about IPS cells. And the prime minister and the politician and everyone loves IPS cells. And so the law about the regenerative medicine has been changed very quickly, more than I expected. Within 3 years, we made a new law for the regenerative medicine. So we have reached to the clinical trial, and that was focused from many countries. And at the end of the 2013, I was chosen for the 5 in the Nature. At first, I was happy I was chosen in the Nature, but if you look at the list of the 5, you can see the Intergovernmental Panel on Climate Change, or maybe you know him, Indian Space Research Organization. So rocket launcher is listed. So I realized this is not the praise, but they think this is dangerous. So we should watch those 5 people. And I was watched whole year, but luckily we did the surgery successfully in that year. And at the end of the year, we were chosen for the 10 who mattered this year. And you can see this rocket person is also chosen. So I was happy for him. We could do the first clinical application. There are 3 reasons, I think. One, that we chose the right cells. We know from the beginning RPE is a very safe cell. We never see the metastatic tumor from the RP cells. And also we can purify them by color. That's very important. If the iPS cells or proliferating cells are contaminated in the graft cells, it will make the tumor. So it's important to purify the cells. And also, the second reason is abduction way of the thinking. And I was told from someone, your thinking way is a little bit different from other people. Usually people think, we did this and the current rule is like this, so we will do this. But your way of thinking is like, you think from the future goal. And I think it's true. So in my mind, from 20 years ago, concrete image of the ideal retinal cell therapy was there. So from there, I can prepare many ways parallelly, and we can judge this rule is good or not. So sometimes we change the rules because nobody knows what kind of iPS cells can be used for the human. So we do the research and also making the rules. So the iPS cells or science has such a big power sometimes. And the third reason, this is the biggest one, as I said, we have a new law for the regenerative medicine. And especially, we have 2 tracks to do the application, clinical application. One is clinical research led by academia, and the laws are different. Law is different from clinical trial. And the clinical trial is the usual way, led by company. But the cell therapy is completely new field of treatment, and this pharmaceutical affairs law for the small molecule drug is not suitable for the cell therapy because cell will change in the body, and we cannot control the cell completely. So we made this change in the law. Also, you should think about the strategy of the regenerative medicine. There are several strategies. One is that drug will stimulate the intrinsic stem cells and regenerate the tissue. This is the most ideal treatment. But usually the disease is too severe that the intrinsic stem cells cannot fix them. Then we should transplant cells from outside. For the cell transplantation, also 2 strategies. One is replace the damaged cells with the regenerated cells. or the second one is trophic effect. And nowadays, there are many clinical trials of the cell therapy, or that called regenerative medicine, but most of them are trophic effect. But in the ophthalmology field, cornea and the retina, we are aiming the replacement therapy. So the requirements of the donor cell is more severe than the trophic effect. And also, we should think about the method. And the surgery and the injection of the cells is completely different treatment. And if you use the intravitreal injection of the cells, it's like a small molecule drug. But if you use the surgery, it's completely different. It's a surgery. So if you think about the cataract surgery, 30 years ago, it was so dangerous and effect is small. But after the accumulation of the effort of ophthalmologists, nowadays, 30 years later, the cataract surgery is very safe and effective. So the regenerative medicine using surgery is also like that. People tend to think the retinal regeneration cures the sight and they can see immediately, but it's not. So at first, the effect is very small, but I'm sure in the future the regenerative medicine will cure many diseases. But usually people think regenerative medicine will be like this. So it's important to avoid the hype and advocacy to the public about the regenerative medicine. That's another important part. Okay, so about the eye. As you may know, the retina is located in the backside of the eye, and the light goes through the transparent tissue like this, and the retina perceives the light. And first, the photoreceptor receives the signal and convert it to the neural signal and transfer it to the bipolar, the secondary neuron, and the tertiary neuron, and the axon of these ganglion cells goes to the brain. And we target these photoreceptor cells and also retinal pigment epithelial cells here. Both of them are very important, and the RPE maintains the photoreceptor cells, so this is also very important. And the human retina has the fovea in the center, and human can see clearly only in this small area, and other part you can see only 20 to 100 or so. So this is a very important part. And the age-related macular degeneration that caused by the senescence of the RPE, retinal pigment epithelial cell, damage this center of the retina. So the people cannot read or see the face. You can walk, but you cannot see the fine things with this disease. And the neovascularization from the outside of the retina cause the damage of the photoreceptor cells, and this part the function will be dropped down. So for this neovascularization, we have a very good drug called anti-VEGF drug injection. But this is not a radical cure, so recurrence occurs often. So patients should receive this injection into the eyeball every 1 or 2 months. And sometimes more than 50 times they should receive the injection. So radical cure might be the replacement of these damaged RP cells. And so for the first patient, we prepare this. From the skin fibroblast of the patients, we made 30 lines of IPS cells, because sometimes it's bad and sometimes it's good. And we chose the good IPS cells and make them differentiate into the RP cells. And again, it's important to purify them, not contaminate of the IPS cells or other cells. And then make the cell sheet and transplant them into the patient's subretinal space. We need the cell processing facility. And the cost of the treatment we should also think. And the most of the cost comes from this maintenance fee of this room. I'd like to do something for this. Cost down is the most important part of the regenerative medicine. But now, still now, we need this firm facility. But it's easy to prove this was RP cells, because RP cells have many functions, and if we check those functions, we can tell this iPS-derived RP cells is authentic RP cells. So we evaluated the specific markers for the RP cells, and we chose the basement membrane in the seed, and we evaluated the RP function. For example, the secretion of PEDF, growth factor, to the apical side, and the VEGF to the basal side. And we confirmed these iPS-derived RP cells is truly similar to the naive RP cells. And about the iPS cells, the safety is the most important issue in the world. Still, people think iPS cell is a very dangerous cell because they proliferate limitlessly. But as I said, RPE is very safe cells, but I should prove that for everyone, not only scientists, but the regulatory authorities and other people. So we did many experiments using various type of cell lines, because the stem cells, even though if it's a one cell line, or even in this colony, we have a heterogeneity here. For example, if some cells may have the gene mutation and others are not. And during the culture, the dominant cells will be changed, or the gene mutations occurs during the passage. So the important thing is we can make the end product the same quality of end product always. So we did this process of purification. And with this process, purification, we can cancel the difference between the cell lines. And we should confirm that. So first we did the single cell RT-PCR and of 200 cells to prove all those cells are really RP cells. And also we evaluated the RP signature genes expression pattern with microarray. And you can see here, those 10 lines of iPS-derived RP cells from different patients shows the similar pattern, expression pattern of the RP cell gene, even though they are completely different cell lines. And also confirmed by the cluster analysis of gene expression profiles, and the transcriptome shows these iPS cells or RP cells in our hands shows the very similar characteristics. We did episomal analysis, and you can see the here. We compare the original fibroblast and the iPS cells and the iPS-derived RP cells in vitro and also in vivo. And you can see that the in vitro RP cells is similar to the fetal RP cells cultured in vitro, and in vivo RP cells is very similar to the RP cells from the eye bank eye, in vivo eye. And this was the episomal specialist says first, this is a pattern of the cancer cells. But finally we found this is just an in vitro character, even though the specialist is saying that this stage, not many people know about the iPS cells or this in vitro change. So many people worried about the safety of the cells, but we did many tumorigenicity tests. And we proved if we use these so-called bad IPS cells, and before the purification, we saw the tumor formation from these cells. But once we purify the cells, we didn't observe any tumor cells in here. So finally, we persuade the committee member and went into the clinical surgery. And again, the purification is very important because here, sorry, this is Japanese, but there's a one cell line, IPS cell line, that have some population of the cells have the B core gene mutation, oncogene mutation. And this is the ratio of the cells that have the gene mutation. And you can see here the blue, 36% of the cells have the gene mutation. And during the maintenance culture, the population is increasing like this. And once you start differentiation, a little bit decrease, but it increase like this. Then you find after the purification, 0% cells have the gene mutation. That means if you have the contamination of bad cells in the end products, you can avoid them with purification. And this kind of phenomenon is different from line to line, so we need the cancel process of the so not only differences among lines, but variety exists in the same cell line with the same protocol. So we cannot yet control cells perfectly. We should be modest that we cannot make the perfect cells. But the important thing is the robust protocol that will not make tumor, even though they have a subtle gene mutation. And we made the first protocol that is very robust safety. So this is the cell that we transplant into the patients. We also should make device for the surgery. And autologous transplantation work or not, we confirmed in monkey. And we did the autologous transplantation of the monkey iPS cell-derived RP cells and also the allogenic transplantation. And we confirmed using the autologous transplant cells, we do not see any immune rejection. But if we transplant allogenic transplant cells, they show the inflammation and edema of the retina, and quickly they are rejected. And we did this surgery, and now 3 years has passed, but the cell sheet remains there without any rejection, and the visual acuity was stable, not improved. From the beginning, we knew it's severe case, so we cannot expect the improvement of the visual acuity. But her visual acuity dropped down from 0.3 to below 0.1, even though she had 13 times injection into the eyeball. So she is very happy after the surgery. Without any treatment, her visual acuity maintained. And her satisfaction score improved after the surgery. But we know that caused by the removal of the damaged tissue. And the authentic true effect of the iPS-derived RP cell sheet is here. The photoreceptor above is maintained after 3 years compared to the other part. And also, we can see the choroidal vessel beneath the cell sheet is maintained. If you see the thickness of the choroidal vessel beneath the cell sheet, this is the transplanted side and outer transplanted side. And if we compare the thickness of the choroidal vessels, you can see here beneath the cell sheet, the thickness is maintained, but other part thickness decreased, atrophy occurred. So the cell sheet works to maintain, they secrete the VEGF to maintain the choroidal vessel beneath, and PEDF to the apical side to maintain the photoreceptor cells. So the cell sheet works after 3 years, I think. Okay, and we published these data in New England Journal last year. And surprisingly, in the same issue of that journal, there's a report about vision loss after intravitreal injection of autologous stem cells for age-related macular degeneration, the same disease. We were surprised, but this is fat stem cells harvested from the berry of the patients. And the in Florida clinic, one clinic operated both eyes on the same day, and it caused the totally blind of these patients. So still this kind of dangerous treatment is underway all over the world, because some people utilize the fame of the regenerative medicine and utilize the hype of the regenerative medicine. So we should be very careful and make the patients cautious about it. So again, the effect of the regenerative medicine is not so high at the beginning. So the autologous transplantation, it shows the immune reaction and also the safety. But it costs very much and time consuming. We can treat only 1 or 2 patients a year. So we move to the allogenic transplantation now. For this, Dr. Yamanaka made another strategy, and he made these special iPS cells that has the HLA6 loci homozygous iPS cells. With this only one cell line, we can treat 17% of Japanese population with HLA matched without any immune suppression. So quickly we evaluate the immune response to HLA-matched iPS cell RP cells in monkey, and we confirmed, even though allogenic, the RP cells suppress the T cell like this, almost like autologous iPS cell-derived RP cells. And if you transplant the allogenic HLA mismatched allogenic RP cells, that stimulate the T cell like this, but if the HLA matched, they suppress the T cell. So we went to we confirmed that in the monkey again and went into the clinical. And this time we used not sheet, but the suspension. And we used these special iPS cells and made the RP cells and transplant them in 2 hospitals. That was the first case of the allogenic in March. But now we finished all planned 5 cases within 6 months or so. So allogenic transplantation, we have tens of patients' RP cells already, so it's more easy and we can treat many patients with allogenic transplantation. And, okay, I should. And as I said, we have 2 tracks. Clinical research, usually the clinical trial will take more than 10 years with $100 million. But clinical research, we can do it very quickly. And from the experience of the clinical, from the patient, we can do the reverse translational research. Again, we need the basic research. We learn from many things from only one patient, and we are now doing the reverse translational research and improve the method quickly. Every 2 years, maybe we should modify the method and make finally ideal treatment of the iPS-derived cells. And can I have 5 more minutes? Okay. So I will talk about the photoreceptor transplantation. This is more difficult because it's a central nervous system, so the cells should be integrated into the retina. And the target disease is a little bit different. Retinitis pigmentosa, whose photoreceptor degenerate because of the gene mutation in photoreceptor cells. And those patients lose their visual field gradually and finally lose the vision. So very desperate disease. But again, the Dr. Sasai, the top basic researcher, developed this method, the three-dimensional culture system from the ES or IPS cells. And they can make the three-dimensional retinal cup from the cluster of neural stem cells derived from ES or IPS cells. So they don't have color, but we can cut out the morphology by morphology and get the purified retinal cells. That's very important again. And we transplant this cell sheet beneath the photoreceptor degenerated mouse. And you can see this, this is a champion data, but you can see the grafted photoreceptor cells remain even 1 year later. And important thing is if they make synapse connection or not. And if we stain this synapse marker, and you can see not many synapses, but some of them form the synapse with the host cells. And function we evaluated in various ways. For example, this is a multi-electrode system in a small dish, and if we put the retina on this multi-electrode, we can detect the response from the tertiary neuron under the transplanted cells. You can see this is a photoreceptor degenerated model mice, so they don't have photoreceptor other side, so they don't respond to the light stimuli. But in the field of transplanted area, you can see the response from the tertiary neuron. So this means the grafted cells connected to the secondary neuron, and they function very well. We evaluated the if it's truly synapse or not by the block of the synapse with AP4 drugs, and if we wash out the drug, the response came back again. So we think this is the synapse reaction. And also we did several behavior tests. This is a shuttle avoidance test. So we stimulate light and sound first and train them. After this signal, 5 seconds later, the electric shock will come. So if the mice learns, they avoid the shock and run into the other room. And without sound, if the mice can see the light, they avoid the shock. So this is a good behavior test, but the result is a little bit difficult to analyze because mouse moved randomly, so sometimes it confused with the effect. So we made the algorithm to detect the blinded mice and the normal mice. And you can see that degeneration model mice shows the blinded pattern like this, but after the photoreceptor transplantation, 40% of the mice shows this normal pattern. So this means photoreceptor transplantation effect afterwards. So we are now doing the monkey model and confirm the functional recovery after the photoreceptor transplantation. And we are doing the safety test to do the clinical trial 2 years later or so. But important thing here is regenerative medicine, the effect is determined by the host environment, not by the cells. So the disease, we should know the disease very well. And if you transplant the photoreceptor too early, the inflammation, microglia attack the donor cells. So we should wait for a while, but after the scar formation, host retina won't accept the synapse from the donor cells. So there's a time window for the transplantation of this kind of neurons. And in the human patients, there are many stages, various stages in one eye, because the photoreceptor degenerate gradually. So this part, just degenerated part, is the good portion to transplant. But again, the effect is not so high. So mental care of the patients is very important. They expect very high effect of the regenerative medicine and come to our outpatient clinic. So it's not. So we should do the mental care of the patients, and we newly made this Kobe Eye Center, which has the research and the cell processing center and the hospital, and also the low vision care in one building, especially for the regenerative medicine and other advanced therapy. And here, actually, we are doing many things, not only the cell therapy, but the patient's iPS cells, and also the low vision care and job support and low vision research, and also the retinal imaging, even auto driving for the visual impaired patients like this. And totally, we would like to help the visual impaired patients in any way. And also, in the future, maybe the iPS cell culture is difficult, I said. Treating them very carefully, we should. And some expert can only maintain the iPS cells very well. But in the future, we will transfer the technique or the skill to the AI robotics. And the life science will be changed with the AI robotics, because you know that half of the experiment is ruined, right? Sometimes. So with the AI robotics or adjusting the good condition, that the waste of time or waste of cost will be reduced, I think. Anyway, this is our experience. Thank you very much for your attention.
Any questions for Professor Masayo? Yes.
It's great talk and elegant lecture. Because you have so many layers of retina. You talked about bipolar cells, outer flexiform, inner flexiform, photoreceptor. Since RP is little away from them, it is it's reasonable for you to graft it in the place. But again, you attempted photoreceptor, but still you could see a lot of formation of synapses with the photoreceptors with other cells. That's how you get the phototransduction. How can you say the possibility of developing all the layers in a lab and transforming the complete sheet there?
Mm-hmm. Yeah, that's that. If we can do that, it's good, perfect. But because we already have the whole layer of the retina, but it's difficult to connect the retina and brain, because the tertiary neuron should elongate the axon to the brain and make the correct synapse with the signal of the location. So that's still difficult. So I targeted the photoreceptor that only inside the retina. So they can just elongate the axon a little bit, several teens microns or so, and connect there is okay. So that's the reason.
Thank you.
Actually, RP and neurosensory retina are part of retina, so it's one layer versus the neurosensory retina. So, and they are interdependent. So it's important to address both.
Professor, I had a question. The therapy that you told us with these iPS cells, can it be used for the brain as well?
Brain?
Yes.
Yes. Actually, in Japan, there are many projects, and one of them is targeted the Parkinson disease, and so they make the dopaminergic cells from the iPS cells and transplant them to the Parkinson disease patients, and it's almost clinical. They are also doing the safety tests, so within 2 years they will be in clinic. And also the spinal cord is another project is for spinal cord injury, and also it's still in the basic research level, but the stroke is the target.
Thank you.
Ma'am, in the case of cells is less, epithelial cell, retinal epithelial cells is less, so that your stem cells induce epithelial cells. But environment of that case in the near retina, the environment that is not progressive for the stem cells, how you make these stem cells will survive there?
Excuse me.
The environment of stem cells where you put these cells in retina, that is not good for these stem cells.
Mhm.
For example, sometimes problem with cells, sometimes the niche surrounding the stem cells is not good. For that cases, how you make this therapy is good?
So we won't transplant stem cells, or because retina is not regeneration. So there's no stem cells. Actually,
Okay.
macular cells is like stem cells, but the not working stem cell is there. So we should transplant the finally differentiated cells, not the stem cells. So we don't need the stem cell niche there. Actually, the ciliary body, the end of the retina is like a niche for the stem cells, but the central part, we don't have the niche.
Even though that case also, for example, niche is not good for the epithelial, that retinal epithelial cells, that may be degenerate after some times, because that is problem with the niche itself.
Yes, niche is important, I know, but the differentiated RP cells or differentiated IP photoreceptors can be survive there. That means the niche or the environment is suitable for those differentiated cells.
Okay, thank you, ma'am.
Any other question? I had a couple of questions. So you said about the tumor form. What type of tumors did they form when they were not selected?
Of course, you are interested in, but we didn't evaluate the pathology really. Usually some project observe the tumor geneticity, but they said not like ordinary tumor. They cannot, even pathologists cannot tell this is what type of cells. And sometimes the neuroblastoma. So not the iPS cell is the problem, but a little bit differentiated, but precursor cells cause the tumor in this case.
Yeah, because the main point of selection is purity, and you said you have confirmed the purity. So in RP, you could confirm the purity because of the visual thing.
Yeah.
How do you confirm the purity in photoreceptors?
So we are now doing, and the purity means we avoid not the single cell type, but avoid the long proliferating cells, and we find the markers to avoid those long proliferating cells. So we will put the immature cell for the photoreceptor transplantation, but we confirm we did many times and we don't form tumor.
Okay, sure. Thank you. Any other question?
Ma'am, actually you're telling that photoreceptor cells are being transplanted. So what day of the photoreceptor cells are being transplanted?
What?
Which day of differentiation?
Which day? Which day? It's a
Because-
good point. It's a secret, but it's okay. We evaluated 60 days differentiation and 100 days differentiation.
Like in vitro.
In vitro, yeah.
Yeah, in vitro, 60 days culture is like they are totally immature, mature cells.
The three-dimensional retinal cell culture needs fully mature retina can obtain after 200 days in culture.
And is there any special technique of transplanting these photoreceptor cells? Like because during transplantation, because these photoreceptor cells, to my knowledge, I know that they have to be transplanted through subretinal space. So is there any means of transplantation, or do you use any cell sheets or scaffolds?
No, the three-dimensional retina has some thickness, so we don't need any scaffold. And we made the device to put the cell sheet or cell suspension into the subretinal space without manual handling. So that's important. So for the regenerative medicine, actually I'm now doing collaboration with various type of company, like a transport company or the cryopreserved company, like not the life science at all. So we should do many things.
Thank you.
This is related to this thing which you mentioned about retinal tissue derived mouse ESC cells, like organoids which we are speaking about. So you are making with the embryonic stem cells, right? But their iPSC are not used. But is it ethical?
So as I said, the good iPS cell is same as ES cells. So we can transfer this technique to the iPS cells easily. But the cell line, if you change the cell line, even in ES cells, if you change the cell line, you should modify a little bit about the differentiation method. So that's a difficult part.
So this one which you are using, it's from embryonic stem cells. Did they try with the induced pluripotent stem cells, like the organoid formation with
iPS cells, you mean?
Because here it's mentioned about ES cells.
Yes, but the
Is it
same
with,
method is applicable for the iPS cells.
But
So you want to know whether they had such kind of
Study
organoids
done
have been derived by IPS by you?
Ah,
Have you derived these from IPS as well?
Yes.
Okay. Thank you, ma'am.
Any other questions? Okay, if not, and this is something that, it's an enviable thing that people can go into clinical phase with the cells that they have derived. And I compliment you, Masayo, for your perseverance.
Yeah.
And that's the key to your success. Thank you very much for taking out time for us. And I would now request thank you. I would now request the PRO office, our public relations office, to do the honors, and our Vice Chancellor is here. So I request you to kindly present the memento and the next one. Thank you.
Wonderful lecture. I joined in the middle. I could join from the beginning.
Now, may I request Dr. Nagaraju to propose a vote of thanks.
Thanks, Dr. Geeta. I think now we are in a stage where the light is about to be there, but it is not there. I think though they are struggling too much to make the cell alive and replace, but ultimately what matters is whether patient is able to see and how well they see. Because usually when we refract the patient after retinal surgery, we also will be very unhappy because it's very difficult to improve the vision, unlike cataract surgery. So I hope cell therapy works much better than the retinal surgery. and all the best to you. Okay. And thank you so much. And I am here to end the session, and it's my privilege to give the vote of thanks. Thanks, Professor Padule Apparao Garu, to honor this session, and we miss you though in the beginning. And thank you, Masayo and Professor Prakash Babu. And I thank the Dean's Office for circulating the information and sending the details to different people. And thank you for being here. And also I thank PRO Office for their efforts to put up the information in the web and also the other details. And I would also like to thank the staff of the Auditorium School of Life Sciences for their help with the AV and the logistics. Thank you so much. And it's my privilege. I think we all enjoy, not I think we all enjoy your talk. And it's really recent update. I'm sure it might have sparked many brains here, the young brains. I'm sure they will try to continue and do some great work. You are an inspiration to us. Thank you.